So to open this forum, we have our wonderful RepleniSENS group lead, Dr. Abdelhadi Rebbaa. His presentation is titled Chemical and Cell-Based Approaches to Reduce Functional Decline with Age. You can read his bio and presentation summary on our events page. And let's welcome Dr. Rebbaa to our stage. Hi, Hadi.
Thank you, Maria, and I would like to thank the entire organizing team, and also thank you all for joining. Today, I will be sharing with you some finding that we are doing in terms of trying to reduce the damage and functional decline with age. Please bear with me. I think my things is not. Because the slide does not move for some reason. Sorry. We do it some other way. Maybe this way. There you go. Okay. So as you know, we are exposed to a variety of damages throughout our life, and they range from environmental damages to metabolic damages to diet-induced damage, and they all lead to molecular and cell and tissue damage that culminate into the induction of chronic diseases and ultimately to aging. Of course, there are some that we can probably control to a certain extent. Maybe we can try to not to be exposed too much to the sun, or live away from a nuclear plant or an area where it's less polluted, or reduce diet and that is harmful. But there are some others that we may not be able to really to control that much. Mostly the damaging agents that actually are within us. And I think, in my opinion, those are the one that needs probably particular attention in terms of research. So at the cellular level, or also at the organ level, damage is known to accumulate in senescent cells, and these cells actually accumulate themselves with age in the body. And these cells are called senescent and defined as irreversible proliferation arrest. So they stop proliferating and they accumulate damage. They are considered to be harmful because they secrete pro-inflammatory and pro-fibrotic factors, and therefore they represent an attractive target for anti-aging interventions. But if we really think about it, there are a lot of other cells also in the body that are also exposed to damage, and I think this should also be considered as a target for anti-aging interventions. Just to put it in this way, every tissue in our body is collection of cells with different phenotype, different age, and they are exposed differently to damage. Some are like deep into the tissue, some are in other area of tissue accessible to damage, and so the amount of the damage that they accumulate is basically depending on their location, on the time of exposure to the damage, and the dose or the amount, the level of the damage. The other thing actually that counts is the ability of the cells to repair the damage. The cells that are young, like stem cells or tissue-specific cells, they can proliferate. They have their enzymes, the damage repair enzyme very active. They can remove damage. They have the proteasome active, lysosomal protease active. to remove aggregate and all these molecules. So and also they can proliferate and they can dilute the damage. Senescent cells, they have their enzymes are not very active. The lysosomes is inhibited, and in addition to that, they cannot proliferate and they cannot dilute the damage. So with this in mind, the senescence actually itself can be defined as the inability of cells to repair damage. So I think that's probably better the definition than just proliferation arrest, because there are cells that do not proliferate in the body. For instance, neurons, they don't proliferate, but the young neurons can repair damage, and the old neurons apparently they cannot. So I think another example is basically red blood cells. Same thing, they don't proliferate, but the young one can survive and they're functional, and the old one basically they just die. So now with this in mind, if we want really to reduce damage, one way was the most evident one, of course, to focus on the cells that accumulate the most, which are senescent cells, and try to target them directly. Since they are already non-functional, even if we remove damage, there's a lot of other things that we need to do to make them functional. So it's probably the easiest way is just to eliminate them directly by using senolytic, for instance. Now, for the other cells that are functional, that are working, and they also have a certain amount of damage in them, but they are fine with it because they're still functional. They are doing their function. But all what we can do for those is to try to not allow the damage to accumulate in them too much until they become senescent. So one way to do that is to reduce a damaging agent that may affect them, or to try to time remove damage from them as it accumulates, to prevent it from reaching a threshold that induces senescence. Now, even better is to try to partially reprogram these cells, because now the technology exists. So this can be even a more compelling approach to partially reprogram them to an early stage. The reason is that these cells, the young cells, they can repair the damage by themselves. So that makes it even more compelling. So of course, the best way is to try to combine one or two of these approaches to really minimize damage to maximum. And that's what we are doing in our RepleniSENS program. We are targeting senescent cells by elimination with senolytics. We are also trying to reduce damage and for the cells who will not even become senescent. And we are trying to partially reprogram functional cells, so they will be able to repair the damage by themselves. For the sake of time, I will focus only on the damage reduction part. And so for this, as you know, senolytic field have taken last few years, and there's a lot of senolytics, mostly for the discovered from cancer. The most common is navitoclax, but most of these senolytics, they target only one pathway and or one molecule in that pathway. And so they're not expressed in every tissue. And in the case of navitoclax, for instance, the target BCL-XL is expressed in other cells and even in the platelets, and that can cause thrombocytopenia and side effects. So this represents a major limitation for using this kind of drugs in the clinic. There's another way to go around it, is to make a prodrugs of navitoclax, and a group in Spain, they add a molecule galactose to it. That galactose actually is substrate for the enzyme that characterize senescent cells, which is senescence-associated beta-galactosidase. And so when it is in cells that do not express this senescence-associated beta-galactosidase, that prodrug will not be active. So even in the platelet themselves, it will not be active. It's active only in cells that express this senescence-associated beta-gal. But we tried in vitro and it works fine. It does actually have a preference for senescent cells, almost like navitoclax. The only thing is that when you try this in vivo, it's toxic also. And the reason is that beta-gal, this enzyme, is expressed in virtually every tissue, and it can be activated by even a small variation of pH. So yeah, it may not be translatable to the clinic also. So we are now working on a different type of prodrugs which are activated by completely different mechanism of action, and we are making progress on that, and I hope I can give you more update in the next time. But just take you back to this problem that actually the Asian research field have, is that there is the lack of markers and the lack of targets to target senescent cells or even to identify them. And this is it has been a problem for a long time, and there's a lot of studies that has been conducted to compare the profile of senescent, non-senescent cells. A lot of omics has very big data studies, but with all this, we don't have a marker yet. And the only targets that actually are targeted by synergics, they are expressed in only a few type of cells and few tissues. So they're still unlikely really to affect, to benefit the whole body. So now, if the goal is to listen or to lower the damage accumulation in the body, for instance, let's say by removing senescent cells, which are actually damaging agent by themselves because they secrete cells, then removing a damaging agent molecule that is endogenous, basically endogenous toxin that is produced within us, that if we can remove that one, then maybe this can lead to the same end, to reducing damage in the body. And one key molecule that it's damaging also, that has been largely overlooked for its role in aging, is heme. As you know, heme is a molecule that is very important for life, for breathing. It is associated with hemoglobin, which is found in red blood cells. And hemoglobin and heme, of course, play a key role in oxygen transport from the lung to the rest of the body, and it plays a role in many areas of metabolism, mitochondrial function, energy production, and many enzymes that utilize heme. However, heme is very toxic, and if it is not well controlled, it can precipitate the end of life. And there are examples for that, which is hemolytic disorders and hemorrhagic crisis. We have found that for the first time, I believe that heme is increased in senescent cells, and we use two different assays, and they all show similar results. Here, this is a kit from EBICAM, and this is a probe that we have identified that is highly specific for heme, for labile heme, and we found that it is increased in senescent cells, and whether endogenous one or if we add heme, is also they retain it, which is very interesting. So for us, this initiate a whole series of investigation, and we're asking a lot of questions that we're investigating right now. First, of course, where does heme come from, and is it toxic? What are the short term of its toxicity? Long term, is it the cause and the consequence of senescence? How to prevent the damage that is caused by heme, And the most, of course, the goal is to, can we target him to treat aging or the associated diseases. And so for this, to give you a little bit where this come from, I want to introduce you to another type of senescent cells, which are senescent red blood cells. They are actually the major source of heme in the body. Each RBC contain about 280 million molecule of hemoglobin, and if you multiply that by 4, which is because every hemoglobin contain 4 hemes, so almost 1 billion of heme molecule per red blood cell. Red blood cells, they live about 120 days, and then they become senescent, and then they go the process of degradation that I will discuss later. So on the daily basis, there's almost 20 billion senescent cells that are cleared every day, and that's a lot. That's really a lot of senescent cells. So now, if we compare that with other senescent cells, it looks like the senescent RBCs are the largest fraction of senescent cells in our body. And also they release heme, which is much more toxic than SASP. And another particularity which is interesting is that they are more dangerous when they are dead than when they are alive. So that means senolytics should not be, of course, used for RBCs. And by this way, actually, the senolytic term needs to be a little bit modified or revised, because it should specify senescent cells that are non-RBC. Other, if it is senolytic for RBC, then it's not good. So this kind of comparison between two senescent cell type bring a lot of question that actually needs to be addressed. So in a normal situation, when red blood cells become old, it gets taken care of by macrophage, and it's recognized, and it's get on the phagocytized, and the heme with it is degraded, and everything is controlled, and the iron go to ferritin, and everything is under control. But there are cases the hemolysis can occur only also by itself, and when macrophages are overwhelmed, and this lead to hemoglobin, and whether tetramer or dimer, but and then of course ultimately to heme. But fortunately there are some protein in the plasma, such as haptoglobin, that take care of hemoglobin dimer, and hemopyxin, which take care of labile heme, and then bring them to either macrophage or to Kupffer cell in the liver to be degraded. But these protective mechanisms become overwhelmed during hemolytic diseases or a hemorrhagic crisis, and this, of course, lead to serious consequences. Now, there are examples of hemolytic conditions. For example, sickle cell disease, where basically hemoglobin is mutated, and red blood cells change morphology, and that can accelerate actually their hemolysis. And basically, these patients, they don't live more than around 52 years. So their lifespan is shortened because of this excess of labile heme in their system. As you can see, the normal amount of labile heme is about 10 to 20 micromolar in the plasma, but then it becomes 50 in intermediate, and then high, more 100 or more micromolar in the disease patients. There are also hemolysis that can happen, like intraventricular hemolysis in preterm born infant, and same thing. The level of heme in their CSF, it's way too high in first few days, but then it regresses, and the survivors they have very strong sequelae that stay with them their entire life. So whether it's chronic exposure to heme or temporary, there are consequences that can stay for a long time. There are, of course, many causes of hemolysis, not only sickle cell, but malaria and hemorrhagic stroke, sepsis, a lot of other things, even viruses and infection, and certain medications that they also induce hemolysis, including the pain medication that we take very often, some antibiotics and environmental factors, airborne particles, smoke, pesticides and insecticides and all kinds of things. So you can imagine, even a normal person, like a healthy person, we are exposed to over at least to the one that are highlighted in blue here, kind of frequently during our lifespan. So every time there is extra hemolysis, that damage continue to accumulate in the body over time, and it can be harmful at the end. With regard to hemolysis with age, there is not much has been done really so far, but this paper, for instance, they talk about the exhaustion of macrophage with age. So they cannot, their ability to phagocytize the red blood cells diminish. Therefore, red blood cells, they just burst in the blood, in the vessel, and release labile heme in the circulation. And right, you can see that in this study, this showed that aged mice have more labile heme in their blood than the young one. And as you can see the value, which is about 50 micromolar in the ages. That's the same, almost the same one that we show here in some patient with hemolytic diseases, suggesting that as we age, probably we will probably start developing this same phenotype as this hemolytic patient. Another thing is that hemolysis do not happen only with age, but it can happen throughout our lifespan, even when we are young. Because this paper here, this show that actually hemolysis can happen just by sheer stress. Just the cells bind to the matrix of the spleen, and just by sheer stress, they can release their heme into the bloodstream, regardless of whether macrophages are exhausted or not. So yeah, all this lead to the accumulation of heme and the associated damage throughout life in the body. And heme, it is there's a lot of literature that shows that it's very toxic and harmful itself. So it does it in many ways. The first one is basically it can contribute to the lysis of RBCs themselves. When there's more labile heme outside, heme is lipophilic, so we just go and bind to the plasma membrane and lyse this more red blood cells. So it creates more like a cycle. If in the vessel it goes to binds to because it's lipophilic, it binds to LDL and oxidize them, and as you know, LDL cause atherosclerosis and other things. It can bind. It can induce platelet aggregation. It can inflammation. It can just bind to any cell in the body, and because lipophilic, it goes to the plasma membrane and there's a lot of RAS and lipid peroxidation, and that causes a variety of damages into the cell. We have shown that in vitro heme can induce proliferation arrest, can even induce senescence, and by comparison with iron itself, at the same amount, heme is more toxic than iron. And we have shown that not only with the cells, but also with activation of probes, with other things, always we found that heme is more potent than iron itself. This is another cell type, which is UVEC endothelial cells, which are actually the one that in vivo are the most exposed to heme. They are very sensitive to it. They can stop proliferating. It can induce inflammation molecule in them very quickly, like AR1 beta here. And if you incubate it a longer time, it induces senescence in them. So it is toxic and can contribute probably to age-associated disease based on this finding. So of course, there's a lot of literature that shows demonstrate toxicity of him. But the question is what to do about it. Can we target it? If we target it, can we reduce these diseases, whether short-term or long-term effects of him? And that actually require, if you want to target it, we need to have a good sensitive method to detect it. And we have developed one. We have found this probe that is highly specific for him. As you can see in the blue bars, the peak is about 0.1 micromolar. When in comparison with iron, the peak is not even obtained even after 5 micromolar, which is like almost 500 times the peak for him. So we use this specific assay to screen for FDA-approved drugs to see if we can find some molecules that can scavenge him, that target him. Before the screen, we optimize the assay, and also we wanted to know, because we will be using him for many weeks, and during the screen, we want to know whether the stability of him is good. So we used one sample that was thawed a few times, and we were using it for a long time. Another one fresh, and with old him with the probe or new him with the probe, they all work fine. And old him alone, no signal. The new him alone, of course, no signal. The buffer, no signal. The probe, no signal. Only when we incubate the him with the probe that we see the signal. And the signal to baseline ratio was very good, so we moved and we start screening. Right now we are halfway. We have another more than 1,000 to go, compound to go, but we already having a good selection of hits, and we're in the process of validating them in an orthogonal way, completely different way. And here I just show you some top candidates, but we have a lot of options actually. This is, you can see the control left, the cells not treated with anything, and the second row, the red one, are treated with him. So there's a lot of damage, as you can see, cells damage, and a lot of them are dying. But when we treat them with any of these 4 drugs, they don't die. So it's really protected. And that's very, very encouraging. It was a good finding that we have made, and I think this drug will have applications for the treatment of hemorrhagic diseases, hemolytic disorders, and also chronic diseases that come from this hemolytic disorders, or that accompany aging itself. The good thing is that these drugs are all FDA-approved drugs. That means their safety profile is well established, and so we don't need to go through all that to prove it. So if we can prove this protection in mice, in animal model, that means it can be translated fairly quickly to humans. So that's the next step that we will be trying. One of them is to try these compounds on sickle cell disease model. These mice, they live about 9 months. 9 months, sorry. So that's a good age that we can do experiment, and hopefully we can extend their lifespan. And then we will also try it in a natural aging model. We will use mice that are about 18 months. And if it's validated in this mice, then as I mentioned, these drugs are FDA approved, so they can be translatable to human fairly quickly. So that will be the next step, the ultimate step, of course. Yeah. So with this, I want to conclude that labile heme, whether it's a result from physiological stress or stress-induced or pathological hemolysis, it is a potent damaging agent that contributes to the onset of progression of chronic diseases. So the development of approach to reduce labile heme or its damaging effects may have application in not only for treating crises, but also aging diseases. And if they approve drugs, that's the one we're screening, that protect against heme damage, may have application and can be easily repurposed to treat aging-associated diseases. And with this, I would like to thank the SRF donors and project sponsors, the SRF board and the senior team for all the help, and the research colleagues and all the entire staff of SRF, of course. And last but not least, a wonderful team of members that are really working very hard to make this possible. And thank you very much. And if you have any question, I'm ready. Thank you.
Thank you, Hadi. That's wonderful. Thank you for the amazing work you're doing for us and for everyone out there.